Again, if you know how the nervous system works, the nervous system has all these cells that have these little channels that allow salts to flow through. They're selective channels. And what you do to generate a current in your brain is allow salt to flow through the membrane. Positive charges, negative charges moving through the membrane, that represents an electrical current. These animals have all of these channels that we have. Sodium, potassium, calcium, etc. They're all channeled in this creature with only 4 cell types. And remember, when I described those 4 cell types, I didn't say any of them was a brain. They do not have a brain, they do not have a nervous system, but they have these little pieces of it. What we think they're using the currents and voltages for, which is probably pretty likely, is they just use it for salt balance. Shuttling ions back and forth is a way to keep yourself from exploding if you become hypotonic or hypoosmotic. So they've got these channels to regulate that, and we've just co-opted those and used those to generate ion flows. They also have complete precursors to synaptic machinery. Again, if you know anything about the nervous system, you have those little wires that got the currents going through them that generates the electrical flow, and then at the end they've got things called synapses which deposit chemicals, neurotransmitters. So you release chemicals there. These are done with vesicles. They have all kinds of little protein machinery in there. They have all the precursors to that machinery in trichoplax, but no synapses. One possibility that needs to be tested is they may be using that machinery as part of the secretory apparatus to release the enzymes that's using to digest its substrate, which sort of implies that maybe your brain is just an overgrown stomach, and that's what we do. Okay, so we find all these precursors there. So, like I was saying with the Big Bang, we've got these remnants that are still floating around there. They're still viable organisms, these protists swimming around, that are carrying little bits and pieces of the machinery that evolved before we developed multicellular animals, and that we have co-opted and used to build our complex structures. So one lesson is evolution reuses everything. And what it does is it repurposes things to fill new jobs. So the question then, of course, is does this mean that evolution can't create anything new? And that's not the case. There are many cases we know where brand new structures have emerged in creatures, and I'll mention just a couple of them, a couple of very simple examples. This one you can find described in Sean Carroll's book, Making of the Fittest, which I recommend highly. He's describing a number of things in that book, but in particular he uses the example of the ice fish. Ice fish are Antarctic fish that have a special situation they have to confront, and that is that the Antarctic waters get very, very cold. They actually get below the freezing point of fresh water, but they don't freeze because they're very salty. So if you're living in the Antarctic, swimming around, you can be swimming in water that is below the freezing point, and fish also have an internal concentration of salt that is lower than the surrounding salt water, which means you could be swimming in water that will freeze you solid. You've got to have an antifreeze to prevent this unfortunate thing from happening. The fish have evolved an antifreeze. This is a new protein. We don't have this protein in us, just the fish. And what this is a hideous diagram from the original paper. And I tell everyone, if you're a scientist and you're publishing, please do not use salmon backgrounds for your diagrams. You know, get one of those arts people to take a look at your pictures, because you could make them better. Anyway, I have some aesthetic disagreements here. What you see at the top is a simple diagram of the gene for the antifreeze protein. And the antifreeze protein is a very simple thing. It's a threonine, alanine, alanine. It's just a tiny little molecule. All it has to do is bind to an ice crystal and prevent it from nucleating the growth of further ice crystals. So that's what the antifreeze does. So you need this very simple molecule. And what you find is, in blue up there, those are the threonine, alanine, alanine repeats all over the place. What you also see in this diagram is there's a stop region, and over at the beginning here, there's some complicated stuff going on. There's a sequence there that is, it's a regulatory sequence. That's where proteins can bind to switch this gene off and on. Because obviously you don't want to turn on the gene everywhere. You don't want it turned on in the middle of your brain. You want it turned on in other places. So there's regulatory regions there to control the expression of this gene. Well, when they looked through the genome to find out where this thing came from, they found a couple of things. One is the second diagram there. That's a trypsinogen-like protease gene. This is a gene for an enzyme, a simple protein that helps digest other proteins. The little orange bits are the active parts of this particular protein. Where this is produced is in the fish's pancreas. And you also have this. You don't have an antifreeze, but you have proteases. And many of them are produced specifically in your pancreas, and then they're secreted into the intestines whenever you eat. And so when you eat a hamburger, well, maybe not a hamburger because it's mostly fat. When you eat something that's meaty, there you will secrete this particular enzyme and it'll help break it down. It's also got a regulatory sequence in front of it. And what they did is they compared these and they discovered that obviously the enzyme is very different from an antifreeze protein, but that regulatory sequence is pretty much the same with a few little differences. It looks like a copy. And furthermore, when you look at the enzyme itself, the first three amino acids in the protein are threonine, alanine, alanine. Oh, this suggests an easy hypothesis how this could have happened, is that there was a gene duplication event. I mentioned this before. It's an easy way to increase the complexity of the genome. There's a gene duplication event that produced a copy of the enzyme, and then there was a mutation that broke the enzyme at that particular point. So it's only producing a threonine, alanine, alanine instead of the whole enzyme. We've got further reason to believe that that's the case because, for instance, this rest of it, this messy thing down here, there was more than one duplication. This enzyme got duplicated a couple of times at different stages in evolution. And when you look in the icefish genome, you also find an intermediate stage where it's got a copy, a very messy copy there. It's got a bunch of threonine, alanine, alanines in it, but it's still got vestiges of the enzyme, the orange parts over there. And it's a non-functional, it's a pseudogene, it doesn't work, but it's just recording. It's a molecular fossil embedded in the genome showing an intermediate stage in the evolution of this. And further, when you ask, well, how does the antifreeze protein actually work? It turns out the antifreeze is produced in the pancreas, and it's secreted into the intestine, where it's absorbed into the bloodstream, where it does its job as an antifreeze. So this is a case where we can trace the evolution of a new protein in an organism. Let me give you one more example here. This is an enzyme called nyelinase. You've all heard of nylon. It's not very stylish, I know, but it's an artificial fabric. It's produced in factories. It didn't exist until I think it was the 30s or 40s when it was first developed. And there are factories that make this stuff. And in Japan, apparently, the factories are kind of sloppy because there are these wastewater ponds surrounding nylon plants that are full of this nasty goo, byproducts of the production of nylon, which looks like a horrible place to grow up, okay? You don't want to be an animal living this stuff. You don't want to drink this stuff. It looks really nasty and smells bad and all this kind of stuff. They sampled from these ponds and they discovered that there were bacteria thriving in it. Bacteria kind of thrive everywhere. These bacteria, though, had evolved the capacity to break down and digest the nylon. This is brand new. We don't have a nylon digesting enzyme. Only these bacteria do. Where did it come from? Well, when they look, this diagram here is a plasmid. It's a little piece of circular DNA that you find in bacteria. And on this plasmid, there are multiple genes all over the place. And the ones we care about are the black ones. Those are nyelinase genes. When they look at these, again, like the ice fish, you find multiple versions. It's like you make a copy of something, you tinker with one, but you still keep the old copy around, and you just pile this up over and over and over again. Again, computer science people, you know this. You know, if you're working on a project, what do? You got a whole bunch of copies stored away somewhere, so you can revert back to an earlier stage if you need to. But here they're just accumulating these. And so we can see some of these nyelinase enzymes they're not very good at their job. They don't digest nylon very well. And then we find others that are a little better, and others that are better still. So they're all recorded there, this history of this thing. And we can ask, where did they come from? And the available evidence, looking at, comparing the sequence of this plasmid with other plasmids, is that it arose by a frame shift mutation. That it took a piece of random DNA and randomized it further and got an enzyme out of it. Now you're going to read it in 3-letter segments still, and that means what you'll read is that. And I think we'd all agree that's gobbledygook, that's garbage. A frame shift mutation typically destroys the function of the sequence that it precedes.